It is a privilege and honor for us to host this evening. We are going to have three speakers, doctors honoris causa from the medical school of the University of Szeged. And my understanding is that it was their special wish to have these lectures delivered here in the Auditorium maximum of the Faculty of Arts, because it is a cultic space, if you like. The 1956 Hungarian uprising and revolution actually started here in Szeged, in this very building and in this very hall. And this Auditorium Maximum has since then has been also important in various public events, in connection with various public events. And since today we are going to have Nobel Prize winner speaker, Professor Sakmann, I am proud to say that 10 years ago we also had Nobel laureate speaking here to the general public, Imre Kertész, who won a Nobel Prize in literature, and we also had an enthusiastic audience back then. So again, let me welcome our guests, let me welcome you, and wish you an exciting evening here. Thank you very much.
So thank you very much, and I'm really happy that this event can occur in this building, and especially because we have heard some words about the history. And I take the occasion and thank for the Faculty of Art hosting us, because it's a unique opportunity. So as it was announced earlier this morning, so it was a privilege to have three doctors of honoris causa this year on the presentation of the medical faculty. So it is also a privilege that one of them is Bert Sakmann, who was very interested in Hungarian history, and during his earlier visit he saw this room, and his memories came out, because when he was young he heard a lot about the historical events occurring in Hungary and Szeged. So as we decided, we will have three lectures this afternoon, or special guests and now or special doctors were asked to deliver a 25-30 minute talk on their scientific achievements and maybe their relationship to Hungary and the University of Szeged. As I have mentioned already, we have three doctors of honoris causa, but because not so many of you attended this morning Senate session, I asked colleagues who proposed this title to deliver their proposal again in this audience. So therefore, first of all, first I'd like to ask Professor Varro to give the laudation of Professor Sakmann.
Bert Sakmann was born in Stuttgart. He graduated as a medical doctor at the Ludwig Maximilian University in Munich. He started to work at the Department of Psychiatry and Neurophysiology at the Max Planck Institute. As a postdoctoral research fellow, he worked at the Department of Biophysics at University College in London. When he returned to Germany, he joined again the biophysical and chemical membrane biology group of the Max Planck Institute. Later on, he became professor at the University of Heidelberg. From 2008, he is the head of the research group of the Max Planck Institute of Neurobiology. During his career, he received several scientific awards: the Louisa Gross Horwitz Place in 1986 with his fellow researcher Erwin Meyer, the Gottfried Wilhelm Leibniz Prize 1987, the Harvey Prize and the Nobel Prize in Physiology in 1991 with Erwin Meyer. Bert Sakmann and Erwin Meyer discovered the so-called patch clamp technique for measuring ion channels in excitable membranes that had revolutionary effect on the understanding of physiological processes. His collaboration with the University of Szeged started when he participated as a guest lecturer at our congress organized for the 75th anniversary of Albert Szent-György's Nobel Prize. Since that time, he has often come to Szeged. He had several lectures for the students and the researchers. In 2016, he was invited to become the scientific director of the Szeged Scientist Academy, and he accepted our request. Due to his current position, he regularly meets the students of our university. He coordinates the activities of the Albert Szent-Györgyi students and supports scientific talent care management of the University of Szeged. Please, Professor Sakmann, deliver honorary lecture.
When your foreign minister, Julia Horn, cut, so to speak, the Iron Curtain, and this was, so to speak, the end effect of this particular movement. It opened up the Iron Curtain, and Hungary is back in Europe, and we are free to come to Hungary. And anyway, this is sort of an emotional moment that I can talk in a hall where all this started 61 years ago. Now, I want to talk to you about some of the science that I did later, and the title of my talk is From Single Channel Currents to Cortical Circuits. So it will be divided into parts. The first question that one asks about signaling in the central nervous system is, where is the electrical membrane voltage, how it is generated? Because all signaling in our nervous system or in our body relies upon fast electrical signals, and they are based on current flow across cell membranes. Now, what is the basis of this? The basis of bioelectricity is a cell membrane that separates ions, these are charged particles, from the inside and the outside, and a membrane permeability referred to as P. And it is the unequal distribution of ions, in this case potassium ions, that will generate a membrane voltage. So this is the basis of bioelectricity. Now, can we calculate how big this membrane potential is? And there's a very old relation by a German biophysicist or physicochemist called Nernst, and he found that the size of the membrane potential referred to as E depends for a particular ion species on the logarithm of the relation between the ion on the inside and the outside. And for example, if you have a 10-fold difference, the logarithm will be 1. So you expect a membrane potential of about 60 millivolts, 60 to 90 millivolts, depending on the ion species. This would be for a singly charged particle. So that's the basis of bioelectricity. That's the basis of how our brain works, how our muscles move, and how our glands secrete. Now the question is, how are these signals generated? They are generated by a change in permeability. So the membrane potential will change because the permeability, which is another term which I left out in this formula, is changing. And the question is, how is the permeability for an ion changing? And there are in principle two mechanisms. One mechanism is called a carrier mechanism. The other mechanism is called a channel mechanism, or probably more appropriate, a pore mechanism. In a carrier mechanism, ions will be passed through the membrane on a one-to-one basis. In an ion channel mechanism, there will be large amounts, 10 to 100,000 of ions flowing through the membrane when a channel opens. So they have a very different size you expect for a elementary mechanism, or for a mechanism that underlies this membrane potential change. Now, Erwin and myself found a way to measure this, and the answer to the question on the previous slide is that electrical signals, at least the fast ones in our body, are generated by an ion channel mechanism. So we were able to prove the ion channel mechanism. We also identified structural determinants of these ion channels. These are proteins, and using point mutational or genetic techniques, one can one by one identify the different amino acids that are responsible for the size and the duration of the current flow. So, and the third aspect that is mentioned in the Nobel notation is that it was the first time that one can observe in real time a single molecule, a functioning single molecule. Now, what is an ion channel current? First of all, I would like to show this slide. This is our group in '81, just after we had completed the methods paper. This is Erwin, that's myself, and these are our collaborators, Owen Hamill, Alain Marty, and Fred Sigworth. They were very helpful or vital for developing this technique. Now, how do we do it? The way to measure the current flow through individual ion channel is schematically shown here for a muscle membrane. This would be a muscle membrane sitting in a bath, and you push a glass pipette that is filled with extracellular solution as closely as possible onto the membrane of the muscle. Any current that will flow inside or outside, into the pipette or out of the pipette, will be measured by a simple feedback circuit, which you can see here. And it is known from basically a lot of work on electrical resistor that the current, the leak current, which is the determinant of the background noise, decreases with the increase of the resistance between the pipette and the extracellular solution. The very simple relation. The leak current is inversely related to the square root of the resistance. Now, how do we do this? Or how did we achieve that? This is the experimental situation as we think what is happening. This would be a cell membrane in blue. This would be a tip of a, wait a sec. the tip of the pipette, and these in red you see individual ion channels. So we are electrically isolating a patch of membrane that has a diameter of about 1 square meter, of 1 micrometer. And the density is low enough so that one can create a situation where you can look at a single channel current. Now, what do we observe? If we use this method, we find that the current that we measure with our background, with our feedback amplifier switches between two states. This would be the upper state, that's the lower state, and we have provided evidence that this corresponds to the closed state, this corresponds to the open state. And you can see these are rather short events, elementary events, that reflect the opening and closing of a single elementary event. It has a size of about 4 picoampere. Now, using this, these measurements, one can easily calculate that only a channel mechanism is able to pass ions at a rate that you can generate a 4 picoamp signal during the 4 milliseconds opening time. So the structural interpretation of this is such that we have an ion, we are looking at a single ion channel in red. It's in the closed state. We activate it by a chemical in this case, and we measure when the channel opens a current. Okay, this would be the current opening. That's the current, and that corresponds to the opening of an ion channel, and ions will pass through the channel as long as the channel is open. When the channel is closing, the current is switched off. So what you see here is an event of a single molecule that changes its conformation from the open state, from the closed state to the open state, stays open, and then closes. The important thing is that using the measured parameters, it points to a mechanism that must be a pore mechanism. Now, to give you some idea of how this looks during a real experiment, this is actually when we are looking on the screen during such a measurement. This would be 2 picoampere, and what you will see is opening and closings of individual ion channels. Okay, so this is what you see during an experiment, opening and closing of individual ion channels, reflecting the association of the transmitter molecule or of the voltage sensor with this molecule. Now, in the lower part, gee, what's going on? In the lower part, you see the size of these elementary events is in the region of picoamperes. Now, what is a picoampere? It is a thousandth of a thousandth of a thousandth of a thousandth of an ampere. Ampere is the size of the current you need to run the lights, and you can see that this is a very tiny proportion of the current. The current is in the order of picoampere. What we have been looking at many channels, and the range of currents is always in the picoampere range, indicating that all these electrical phenomena that are generated by the opening and closing of these molecules, or by the action of these molecules, are ion channels and not carriers. However, there are slower phenomena in the nervous system, which are indeed generated by carriers. But the take-home message of this is all the fast signaling, that is, when we react or we make a movement, is based on the opening and closing of these ion channels, and the signals that run our body and our brain are generated by the superposition of thousands and thousands of such elementary current events. Now, this is the first part, and I would like to show you now that once you have a technique that has been designed for a particular purpose, you can use this technique for other purposes, for unexpected purposes. And I want to show you one example. The reason why I chose this is that Gabor Szabo said at the opening of the Eli that this lab is developing methods, and it's sooner or later these methods that will be developed at Eli will sooner or later be used in other fields of science, in particular in biology. And I just want to show you that this is also true for the so-called patch clamp technique. We found an application and made discoveries that were completely unexpected because we found we can use this technique for other purposes. And this has to do with the signaling within nerve cells. Probably most of you know that nerve cells consist of three compartments: a dendrite, that is the receiving compartment, a soma, this is where the nucleus is located, and the axon, this is the sending compartment. And these three compartments of a nerve cell are connected to other nerve cells via synapses. And it has been a long sort of dogma, almost 100 years, that only the soma and the axon are electrically excitable because they have ion channels. Now, we found that these very small structures, dendrites, they have a diameter of 2 to 3 micrometer, up to 1 micrometer, or down to 1 micrometer, can be investigated with the same technique, or with the modification of the technique that we use to measure single channel currents. And this is shown here. The experimental setup is such that we are looking or identifying a particular cell with its soma, its dendrites, and its axons, and we can use the patch clamp technique to establish recording from the soma and from the dendrite. And what we found is that, contrary to common knowledge, the dendrites are electrically excitable. So a nerve cell has three compartments: dendrites, soma, and axon, and all three compartments are electrically excitable. The next question then is, what could be the purpose of this? Why should the dendrites be electrically excitable? And the answer is shown in the next little video. I'm skipping 10 years of work, but what we found is that based on the electrical excitability of the dendrites, a single cell can act as a coincidence detector for synaptic input arriving at different parts of the dendrite. Now, we have enough physicists in the audience, I hope, who knows what a coincidence detector is. Okay, Gabor, what is a coincidence detector? Okay, I'll tell you. In biological terms, a coincidence detector is a device for which a Nobel Prize was given in 1952 that will have an output only when there are two spatially separated inputs within a certain time interval. Let's say 1 millisecond, 10 milliseconds, one microsecond. And this device has been used to look, for example, at cosmic ray, at the origin of cosmic ray. Turns out that in the central nervous system, there is a biological equivalent. A certain type of pyramidal neurons are coincidence detectors for input arriving in different parts of the dendritic arbor. And now watch what's happening in this situation. We are activating two inputs. One is called a dendritic, the other one the somatic. The one is shown in yellow, the other one is shown in blue. And we are now shifting the time where this input is active and this input is active. And what you will see is that out of a single action potential or no action potential, there will be several action potentials once these two inputs are occurring within a certain time interval. Now, I hope it works. Okay. So what you see here is that for two coincident inputs that have a certain time difference, instead of a single action potentials, there will be three action potentials. You have seen this? No. So what is happening is that these cells are coincidence detectors that detect input to the apical dendrites and to the basal dendrites when the inputs are occurring within less than 50 milliseconds. Now, what could this be good for? We have provided evidence that, for example, you have an input through your visual system and your acoustic system, and this will generate a particular reaction when these two stimuli occur within a certain time interval. And the basis for this is this type of cell that increases its output by a factor of 3 when the two stimuli are occurring in a short time interval. So using patch clamp, a modified version of the patch clamp technique, we have discovered that the dendrites of nerve cells are electrically excitable, and the function of this excitability is to endow the cells with the capability of coincidence detection. Now, in the last 2 minutes, I would like to show this more visually, because I was at the opening of the Eli, and as I said, they are developing beautiful techniques. And one example that is, happened maybe 15 years ago, where a new development in laser physics opens a completely new world to neuroscience, is the fact that you can use two-photon imaging to look at the electrical excitability of this type of cells. The basis of this is that the cell membrane of the dendrites is not only permeable to sodium, potassium chloride, but also to calcium. And if you fill the cell with a calcium-sensitive fluorophore, you can look at the optical signal that is generated when an action potential is generated across this membrane using two-photon microscopy, which relies upon the development of short pulse lasers with short pulses. I won't go into the detail. The fact is that using this technique, one can look at the electrical excitability of nerve cells with purely visual means, with a visual technique. And my last slide then is this. what you will see is these are nerve cells that have been filled with a calcium-sensitive dye. We will activate them by a natural stimulus, and you will see when the light becomes warmer, it indicates there are electrical signals, and you will see that these electrical signals are not restricted only to the soma and to the axon, but they also travel back into the dendritic arbor. The big advantage of this technique, as you can see, is you can look at many cells at the same time, which you can't do with electrical means. So this has opened a completely new field of investigating the central nervous system due to the fact that laser physicists have developed lasers that are able to to generate laser pulses in the picosecond rate. This is very boring for Gabor. He's in the femtosecond, but in biology we are right now at picosecond. But I'm sure there will be some application for your femtosecond lasers. Now let's have a look on how this looks. So what you see is, I'll keep this cycling, is cells filled with a calcium-sensitive dye, and whenever there is an increase in the wavelength of the emitted light, that is, when it's getting red, means there is electrical activity. And the beauty of this technique is we can first of all look at many cells, and we also can look at the whole dendrite. Whereas with electrical measurement, one is basically restricted to 2 points. I'll just show this once more again, and then I'm done. I just have to look at this. So, can you see this here? Okay, anyway. Has everybody seen it? Yes. Okay, good. I would like to thank you again for the occasion of awarding me a honorary degree, for the occasion to be able to talk into this lecture room, and for the occasion to show mostly people from the medical field how important it is to have to look what's happening in physics. And this is just one example. It has basically changed the field, and I'm sure, and I hope, that the ELI will create instruments that will be as useful to us as the two-photon microscopy. Thank you very much.